Okay, so um, I have not released the multiple choice scores yet for the exam that we took on Friday because not everyone's taken it yet. So as soon as that's accomplished, then I can release the scores and send an announcement that they're there. Okay. And then I haven't had a chance to look through any of it at all. I do go through each question. I still have to do that for the second exam as well. Go through each question, look at the stats, and see if anything needs to be shifted, points added on or not. So I will be doing that probably over break. Will be when I can get to it. All right. So here we're looking at where we left off, I believe. So we're thinking about how plants are defending themselves, and it really does depend on what's attacking them. Is it a fungus? Is it a bacteria? Is it an insect? What kind of insect? There's all kinds of insects that can attack plants. So it does make sense when you stop and think about it that the plant should have some way to recognize who's attacking so that they can mount a very specific defense. And that's more energy efficient on the plant's part if they can be very specific in their response rather than just trying to get everything up and running to defend against everything. That would be way too costly in terms of energy and resources. So here we're looking at the idea of elicitors and how I think in the previous slide we were looking at that definition of an elicitor where there's lots of elicitors out there, but it's usually something that is coming from the microbe or the insect that the plant is recognizing. And from that recognition, it will mount a defense. So calcium, they were kind of surprised to find out that calcium is a major player. They didn't really hypothesize that to be the case, but when they started looking, they found calcium is heavily involved. And the more they look, the more they find. And so calcium is really important as a second messenger to relay that information to the inside of the cell, shift gene expression, get that signal cascade system going so that ultimately you can get a shift in gene expression. We're going to be introduced to some terms. We already looked at in the last slide, HAMPs. We're going to, here's MAPK, where terrestrial plants now they think have way more of these protein kinases, again, which are involved in signal transduction. It's their way of relaying. In this case, I'm getting attacked and then getting that signal all the way down to the DNA level so that you can get a correct gene expression pattern, hopefully to mount a very effective attack. So let's look at an example here. Let's say an insect is coming along and it's starting to chew on the plant. A couple of things happen right away. Cysteamine is a protein that's actually a hormone. It acts like a hormone. So far, it's the only one that they know, but there's probably others out there. We just haven't found them. So what they see happening is soon after the chewing by the insect, this cysteamine hormone is going to bind into a receptor on the cell membrane, CM cell membrane. And then that quickly gets jasmonic acid going. And so you get this upregulation in jasmonic acid, and it will start to play a role in the signal transduction as well. It also will do a couple of other things. It will activate these so-called protease inhibitors. So these are enzymes that act as anti-digestive factors in the insect. And so as the insect is ingesting these protease inhibitors, it messes with the insect's ability to digest. And if the insect can't digest its food, it's not going to get any caloric intake from that. And it can cause, essentially they're starving. They won't be able to use any of that. And so that can get the insect to move on and try another plant. And so it can be effective. They also see that cysteamine will often ramp up terpene synthesis. So those secondary metabolites, as well as alkaloids. And again, it depends on the insect, which flavor of secondary metabolites will get upregulated the most. And so, but it's the wounding event that is kicking all this off. And so here is a visual of a couple of things going on here. Here's the caterpillar chewing on a leaf. They're saying, okay, that's definitely wounding. And so that will kick in jasmonic acid. You'll get gene expression upregulated to try to deal with that. Volixitin is one of the chemicals that is actually in the saliva of the insect that the plant is cueing in on. And so here it is causing, what you're seeing here is jasmonic acid. That hormone starts to get upregulated. It kicks in a signal transduction pathway that actually gets the plant to release volatiles to attract a parasitoid wasp. And the reason it's doing that is the plant is calling, so to speak, calling to this wasp to say, hey, why don't you come on over here? There's this juicy, yummy caterpillar chewing on me. Why don't you come and get it and eat it? And so it's that idea of the enemy of my enemy is my friend. And so the plant is saying, hey, you know, all you predators that like to eat this caterpillar, come get it. And so it's a way for the plant to ultimately deal with the caterpillar by calling in the predators to the caterpillar.
My parents have a passion vine in Florida, and I literally saw this interaction happening with the wasp trying to lay its eggs.
Ooh, nice.
I didn't know it. I know.
Yeah. 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 So yeah, very interesting predator-prey interaction. And you know, they were shocked to find out when they discovered this. This made big headlines in the plant world because that was just shocking to think that the plant was playing such an important role in getting the predator to come over. And so yet again, you know, plants continue to shock people who don't understand how complicated plants are and how sophisticated they can be. And so the end result is that the predator will come along with that volatile release from the plant. It will follow that volatiles concentration gradient, find the plant that's making it, and find the caterpillar in this case. So no free lunch. So here's another more in-depth look at this idea of systemin and hormones. So here you see the systemin getting docked into the receptor protein after wounding by a caterpillar or something. And then you can see a couple of things happening here. You see that that will kick in jasmonic acid, and then you see that can kick in the proteinase inhibitors, get those up and running so that as the caterpillar continues to chew, they're going to get a dose of those into their digestive tract. And so this is a signaling pathway that they've been working on. So this is taking that previous slide now and showing a little bit more of the detail of what's going on. So here, if you look, this poor tomato plant is getting attacked. They're showing it's like a caterpillar on that leaf right there. And so what they're trying to show is that if you look down to the lower right, you see that systemin hormone getting released, manufactured, released, and then docking into the receptor in the cell membrane, and then that will trigger jasmonic acid synthesis. And then you see they're trying to show that that's part of the signal transduction that will help to get the gene expression pattern to shift into a defense response. And then you also see the jasmonic acid, they're trying to show that it can get sent through the plant. So it's not just on the leaf where the caterpillar is chewing. And so this is another big find, that they were surprised how effective plants can be at taking what's happening in one part of its body, but then alerting the rest of its body, uh-oh, you know, I'm getting chewed on this leaf. We better get the whole plant body ready to defend itself against this particular in this case, a caterpillar. And so what they're trying to show in this drawing is that jasmonic acid can very quickly get transported through the plant body via the phloem and can activate distant cells, distant tissues. And so this is the idea of a full systemic body response, so that here you see the word systemic. So they're just trying to say that this one leaf getting chewed on will send a whole systemic response throughout the plant body. And again, if you think about it, it's like, well, that's really smart. And it's a good adaptation, because obviously these are crawling insects. A lot of them are flying insects. They can easily hop or crawl to another leaf and start it up all over again. So it would make sense that the rest of the body is getting primed to defend itself. So hopefully when the caterpillar goes on to another leaf, it will already have be encountering a good defense response by the plant. So hormone flow through the phloem is pretty standard. That's one way to get a distant part of the plant body up and running. So jasmonic acid. So it can be produced at the damage site and go systemic. They've also been tracking electrical signals through the plant body. That's another way to transmit information. It's not as fast as what happens in our brain tissue, for example. It's not quite that fast, but it is a way to get a signal relayed to distant parts of the body. And I'll talk a little bit more about that as well when we get into it. But here is, they're trying to show that in a visual format here, where they're showing the caterpillar chewing away, the upregulation of jasmonic acid to get defense responses against that. And then you see it's also triggering electrical signals. All right, so that was another big find. They did not at all anticipate that plants were using any kind of electrical pathway, but they now know it's well documented in many different plants. So I'll talk a little bit more about that in a bit here. We also have vibrational signals, which again was very, they just didn't think that this was a thing, that plants could do this. But basically, when you think about an insect crawling on the leaf, let's say, or a caterpillar chewing on the leaf, that does send out, I should say, vibrations, whether it be the vibration of them crawling or the chewing vibration. That leaf tissue can pick up on that vibration, that energy, and they can respond to that. So this is a paper that was published, the first paper that was published on it. I remember I went to a plant biology conference shortly after it was published, and the authors of the paper were there to present their work on how they did this. And it was standing room only. There was overflow into the hallways because people were so curious about this. It just seemed so fantastic at that time to think that plants could do that. And so there was a lot of interest in it. And initially it was met with a little bit of pushback by other scientists saying, nah, this isn't really happening. You did the experiment wrong. But they did a very careful, it was peer reviewed, very carefully done. And then further studies were backing up that paper's findings. So they're actually able to pick up on those vibrations, and that is information to the plant that they're getting attacked. And so it elicits a chemical defense by the plant, and just by the vibration impact of in this case, it was against chewing. And so here you can see, I just put this is from the other slide set, just looking at the glucosinolate. So what they were doing was they did a controlled study in the lab where they mimicked chewing, like the recording of the chewing by the insect, and they clamped that onto the leaf and did the recording, and they were looking to see how the plant responded. So they didn't have, in this part of the experiment, an actual insect, they just had the recording. And so this is data from that plant where they're looking at, so the playback leaf is the leaf that actually got clamped on with the recording, and then the systemic leaf was on the other side of the plant, and then the rosette center is just the dead center of the plant. So the playback leaf over on the right here, figure B, it's PBL, And then what they were showing was that the electrical signaling passed over to the systemic leaf. And you can see that when there's that chewing, it's the asterisks above the bar graph means it's statistically significant. So there's a significant upregulation in glucosinolates by the recording, whether it's the leaf that actually had the clamped recording onto or the systemic leaf a distance away from the plant on the other side of the plant, essentially. And so they were able to show that there is that response by the plant of upregulating glucosinolates because it thought it was getting chewed on by an insect. So it did the appropriate defense response by upregulating those glucosinolates. So then they asked, okay, so, what other vibrational signals, does any vibrational signal get this upregulation in glucosinolates? So they did another experiment where they did the chewing again, recording of that. And then they did a recording of a leaf hopper that's just sitting, like leaf hoppers will sit on plants and they'll do their thing to attract mates. So it was just a mating call that the leaf hopper was doing. So they recorded that and exposed the plant to it. And then they exposed the plant to a recording of wind. And again, those are all causing vibrations. And they just wanted to see, can the plant recognize the truly dangerous one and ignore the irrelevant, non-dangerous ones. And that's what this is showing, that when it was exposed to leafhopper mating calls or the wind, it didn't do anything in terms of, in this case, they looked at anthocyanin upregulation, but chewing, yes. So again, the plant was paying attention to the ecologically relevant vibration and not to any old vibration, which makes sense. If you think about all the vibrations a plant experiences throughout its day, it's a lot. So it has to have some way to figure out which ones it should really be paying attention to. So when this was published, this was back in 2014, it was a big deal because this was really one of the first carefully done studies with proper controls that could show that vibrations can, the plant can pick up on that and use that appropriately. So I think one of the reasons why it was met with some skepticism is that there was a book published, I'm blanking on the copyright, a couple decades before this, that was talking a lot about how plants can sense their environment, and it kept citing it in anecdotal information or non-science information, but it captured the imagination of so many people. And so it unfortunately greatly slowed down any productive research in the area of how plants respond to their environment and how they perceive their environment because of that book. It's kind of an interesting, it was like a big block. People didn't want to get into this research because it was so poorly done in that book that people were afraid to even get into it because they didn't think they could get any research money for it. They thought they'd be ridiculed, and by and large, that's what happened. When people finally started getting into this area of research, there was a lot of pushback, a lot of arguments amongst the scientists. There was even one scientist at a plant biology conference. A bunch of other scientists made a petition and tried to get signatures to get them ousted out of the society because they didn't like the research they were doing. And turns out it was really good research. That scientist did a really good job and luckily kept going and was able to publish. So this was one of the persons that was kind of criticized, Dr. Gagliano, but she's done some really careful studies and with proper controls and has some very compelling data to look at the idea that plants can understand acoustic signals. And so you might have heard, that plants can they can respond to music. You might have heard that one. And that was in that book I was talking about that wasn't very well done. It's not the music, plants don't care if it's Led Zeppelin or classical or whatever. It's the vibrational frequency that they're picking up on. And so if you phrase it in the right way, acoustic vibrations, certainly plants can pick up on. so anyways, there's a lot of really interesting research in essentially the different senses of the plants, that they have their own version of hearing, for example. And so in this case, it's the vibrations that they're picking up on. So what they're finding is when insects in particular are chewing on the plant, you get jasmonic acid hormone is a common strategy. They will produce that right where the insect is chewing, plus they'll send it out through the phloem and tell the rest of the body to gear up and start protecting themselves. Other systemic defenses are the electrical signals that we mentioned, vibrational signals. And then don't forget that plants can also send out volatiles. So if you can imagine, for example, let's say a bigger plant, like a tree that has a multi-story canopy, a huge crown on that tree. What if the lower leaves on the lower branches are getting attacked? You need a way to tell the whole canopy tree, the whole part, to get ready. And so sometimes it makes sense to just have volatiles coming out of those bottom leaves that are getting attacked and have those waft up into the rest of the crown of the tree, for example. And so you may see in the literature, they'll talk about green leafy volatiles. It's just kind of a general term that they use for a bunch of different volatiles that can be used in order to, in many cases, prime the rest of the plant. It's a systemic signaling system so that the rest of the leaves on the plant, for example, can ramp up their ability to defend themselves against whatever's attacking them. So there is, several different strategies that can be employed. It just depends on the situation and what's attacking them, how the plant will work the response. Here, they're looking at a couple of different things. So here you can see that there's some nematodes here. Well, here's the, looks like some kind of caterpillar or grub that's chewing on the plant, perhaps, or I guess it's supposed to be a nematode. That's enough. Nematode. Anyways, apparently that's a nematode. And then, no, it can't be a nematode. These are the nematodes. I don't know what the artist is thinking there. But anyways, what you're seeing here is that the caterpillar up on the leaf is chewing, and it is sending systemic responses now to the whole plant body. And here they're showing how the predator gets attracted by volatiles coming now. So it'll come in and eat the caterpillar. Here you're looking at direct inhibition of other herbivores that might be trying to come along and lay their eggs, for example, onto the plant. So HIPV, by the way, that stands for herbivore-induced plant volatiles. So they just mean that the chewing insect, the herbivore, was inducing that response. And then here you're seeing that the neighbor plants are getting signals from the attacked plant, a volatile typically, in this case at least, where you can see the neighboring plants are starting to ramp up their defenses against that particular insect, even though they themselves aren't getting eaten by that insect yet. And so this idea, again, was huge in the plant world. And again, so many scientists said, no, this isn't happening. That just doesn't happen. And they accused the scientists who first did a study on this that they did it wrong. You didn't have proper controls and you contaminated the samples. And so then those scientists went back and they went into the lab and did a really carefully controlled study, proper controls. They thought of everything, and they still showed that this was happening. And so that kind of those scientists had to shut up and and deal with the fact that there are plants that communicate with other plants in this way. So it depends on the situation. This one was volatile. The original paper I'm talking about, it was a volatile signal. And that's why they said, no, you had too many variables in your experiment. That wasn't. So then, because the first experiment was a field experiment. And yeah, sure, there's a lot of volatiles out in the real world. So then they went into the lab and did very careful, contained, everything was like a plastic bubble type thing. And so they had proper controls. And in that case, they were demonstrating it's a volatile. Yeah. and it wafts over to the neighbors. Yeah. And so this plant essentially sent the right volatile to tell these other plants, I'm getting attacked by this caterpillar. So you're probably next, ramp up. And they followed it out and they took samples from the neighboring plant. They showed the shift in gene expression. And so it was a case here where this plant was telling nearby plants. Now the question is, was that done on purpose or was that just kind of a knee-jerk reaction by the plant, so to speak, and it wasn't really trying? So there's a lot of debate out there on that idea. And then the whole idea of, well, would a plant do that more for its own kin versus non-kin? So they're looking at that idea, and they're showing that plants can detect around them who's kin and who's non-kin. And so there's some really nice studies showing a lot of that can be done through root connections, chemical communication via the root connections or the mycorrhizal connections, but sometimes volatile. So lots of interesting work looking at kind of the sociology of that, for lack of a better way to think about it. But the papers that I've read on it, the data is pretty strong that they can recognize kin versus non-kin, and that they can send out signals to non-kin. Usually when you think about it, you want your own species to survive and persist, right? Why would you help out strangers? You want your kin to survive so you can pass your genes on to the next generation. But they also bring up the idea that, well, you don't want a monoculture either, though. You need diversity in ecosystems for a healthy ecosystem. So even though these non-kin plants are picking up on it and surviving better for it, well, that just may, even though they're non-kin, it may still help out this plant, because generally the general rule of thumb is areas that have high diversity in organisms is usually a healthier environment for everybody in that environment. So lots of interesting sociology and evolutionary questions going on with this. Yes, so mycorrhizal fungi, I was just reading a couple papers late last week, how mycorrhizal fungi now they're realizing can really control the flow of what, like this tree is going to get so much carbon, whereas that tree is going to get some lower amounts of carbon, and this one's going to get more phosphorus and this one not so much. And so they're finding that the mycorrhizal fungus is kind of like a middle person in the flow of energy resources. And so it's interesting. They've done some really careful, well thought out studies. They'll actually insert like these mesh plastic hollow conical things that they'll have the roots in, and then they'll actually like either rotate that conical so that they're breaking any mycorrhizal connections, so that like this plant on purpose, we're not going to make you mycorrhizal, but we'll let the other ones go. And so they're coming up with some pretty simple but ingenious ways to kind of look at this idea of what are the mycorrhizal fungi doing. But now they're realizing they play a big role in this kind of communication as well. This picture here is just trying to show kind of a big picture idea of lots of different options the plant might have depending on what's attacking them. So this is kind of like a review, thinking about some of the things that they can do. A lot of it is VOCs, volatile organic carbons that are getting released. They talked a little bit about the plant microbe interactions in the soil. So the microbes, the mycorrhizal fungi, as well as beneficial bacteria, can influence the plant's ability to help defend itself as well. Okay, let's look at some examples of more in the microbial world. So here you're looking at a leaf, and they're showing how bacteria and fungi are starting to gain entrance. So certainly the stomata, I don't know if I have let me look here. Yeah, I'll show you a picture in a bit here. The stomata is definitely big enough for, the pore is big enough for bacteria and fungi to get in. It's pretty straightforward. So unfortunately, that's one easy way for microbes to get in. but that's also a way for the good guys to get in. So it's not just pathogens that will be doing that. So they're trying to show that here, that there's microbes entering in through the stomata. You can get them directly, like digesting for, their way through the wax, the cutin, and getting through that way just by damaging the cell wall and cell membrane. Here, this potato, if you've ever looked at a potato and you see those little bumps, those are, well, this is one that looks diseased, but a normal potato just has these little tiny like striations. Those are actual, they're called lenticels. I don't know if you can, should have picked a white color for that. Lenticels, we learn about that in the plant kingdom. Those are essentially pores along bark on the stem of the tree. And that brown, the skin of the potato that you eat, that's actually bark tissue. And so those are lenticels, and that's another way for microbes to gain entrance because it's a relatively loose area of cells that they're not very packed together. And so it's a way for bark tissue to have oxygen flow coming in fairly easily. But that's another entry site for microbes. Okay, so and then the reason I have an aphid here is to get you thinking about excuse me, how they have microbes in their digestive tract. When they insert their mouth into the plant, some of those microbes get into the plant. They kind of like almost regurgitated into the plant through the mouth part. And so you can get insects transferring microbes as well. That's a very famous way for microbes to get from plant to plant. They get a free ride with the insect. Okay, so let's think a little bit about how, so far we've been talking about insects. Let's talk about fungi and bacteria. So they will be producing effectors. That's often how you see it talked about when we are thinking more specifically about fungi and bacteria that are acting as pathogens. Effector molecules, it just makes them more effective at being a pathogen. Think about it that way. Effectors make them effective at being a pathogen. So these so-called effector molecules, they can be enzymes that help break down the cell wall, for example, so that they can get into the cell and get those contents of the plant cell released so they can sop it up and eat it. Might be a toxin, a very specific molecule that they make and excrete to act as a toxin. It could be growth regulators that will actually overstimulate a plant to maybe produce a gall or a tumor, so to speak. But basically, an effector molecule, it is making them more effective at being a good pathogen and allows them to attach and colonize, get access to the plant's nutrients and resources. So really, they are a type of elicitor. We already defined what an elicitor is. It's something coming from the whatever's attacking the plant. And so effectors, unfortunately, when you get into this realm of research and study, they have a lot of they like jargon. And so there's a lot of jargon and abbreviations. So here's another one, MAMPs. These are another acronym. Stands for Microbe Associated Molecular Patterns. We already looked at HAMPs with herbivore associated molecular patterns. Now we're looking at MAMPs. So again, it's just something coming from the pathogen. It might be a physical part of the pathogen. That's very common. For example, bacteria have flagella that allow them to move from point A to point B. Well, plants don't have flagella. It's a very foreign thing for a plant. And so they can pick up on that and they can sense that there is something here with a flagella. That's usually something to make them think twice about what's going on. Fungi have chitin that plants don't have that. Plants don't have chitin. So again, that could be a red flag for the plant. Oh, I'm sensing chitin. What, should I be alarmed or is this one of the good guys? And so again, that's an example of a MAMP. So these MAMPs, they're detected by plants. Plants have these so-called PRRs, pattern recognition receptors. So they're just receptors. Often they're in the cell membrane, but not always. And so, for example, the flagella or a piece of the flagella would dock into that receptor protein. And that docking into the receptor protein, that action would wake up the plant, so to speak, and go, look, you got some bacteria here. You better figure out if this is friend or foe. All right, and so there are these PRRs. So typically they're on the cell surface. That makes sense that most of them are there because that's the first line of defense against these insects and fungi and bacteria. So they'll refer to this response by the plant as MAMP triggered immunity. So it's an immune response by the plant because it's using these MAMPs as, they're detecting that and going, okay, I got a flagella here docked into my receptor, into my PRR. I got to figure out what to do with this. All right. So we're going to look at that in a picture here. Next, let me pull that. So here on the left, you're seeing that what I just talked about, where you have MAMPs coming off of the flagella of the bacteria, or maybe there's some chitin cell wall material that is docking into these PRRs, okay, pattern recognition receptors. So this is the idea of once that happens, once you get that MAMP docked into the receptor protein, that's going to trigger a signal cascade all the way, hopefully, to shifting the gene expression so the plant starts to defend itself if it needs to, if it's a pathogen. So that's what the MAMP triggered immunity is getting at. Now, let's see. Let me go back. Let me double check. All right, I'll come back to that picture in a minute. Let me just show you some visuals here. This is, if you can make sense of it, so right where my red laser, that's a stoma right there. So there's two kidney-shaped guard cells, and then the pore in the middle. And then there's another one right here, two kidney-shaped guard cells, and the pore in the middle. So you can see Pseudomonas was stained green here, and you can see how they're easily small enough to fit into the pore of the stomata. So that's an easy way. See, they're on the leaf surface and pretty good numbers just in this little tiny spot of the leaf. And they're definitely slipping in through the stomata. Here's another picture, really nice picture showing that idea. So both good and bad microbes can get in this way. And so now plants can, as we know, they can regulate their stomata, the pore size. They can shut down their pore. And now they actually know that there's bacteria, good bacteria, that can tell the plant, hey, guess what? There's a bunch of these pathogens around me. You better shut down your stomata. And it can actually, so the good bacteria tells the plant there's bad bacteria hanging out and you better shut down. And they can actually, they did a really nice study tracking that whole signal cascade, and then the plant would close down its stomata. So it's amazing how these interactions, the more they look, the more sophisticated it turns out to be. Okay, so that, what we just described with that MAMP triggered immunity, sometimes it doesn't work. Sometimes the pathogen is able to figure out another way and kind of trick the plant. And so sometimes those effector molecules don't get picked up by the receptor protein. And so the plant is kind of clueless. So sometimes what happens is that might mean that the pathogen, either part of the pathogen or some of the pathogen, gains entry into the cell of the plant. Well, the plant has a second line of defense. So inside the cells, there can be what they call R proteins that are floating around in the cytoplasm. And if they encounter one of these effector molecules, then one of the MAMPs, so to speak, then it can cause the plant to still mount a defense. And so they call that idea effector triggered immunity. So the effector could be a toxin that gets all the way in, or something like that, that it's foreign to the plant. It's a non-plant molecule. So going back to that picture again, that's what's being shown on the right. So here, what's going on is I often think of the analogy of these effectors. It's kind of like when you have to wait in line to get into a bar and you have to be on the list, and so, but if you know the back door in, and you can sneak by the bouncers, then you get in. And so I often think of that analogy when I'm thinking about this effector triggered immunity. They got in through the back door and the bouncers never picked up on it. But then once they got in, the bouncers circulating around go, hey, you're not supposed to be in here. All right, so there are these so-called effector molecules from the pathogen, but luckily the plant has these R proteins floating around and they can bind to those effectors. And again, that binding action triggers the plant to go ahead and mount a defense. And so here you can see they're trying to show that signal cascade and the effector triggered immunity. So the plant has more than one way to deal with this. And again, that makes sense when you think about all the different pathogens and things that can attack the plant, that they should have some sophisticated immune system in play if they're going to survive that. This isn't from your book, but I thought I'd put it in there anyways. It kind of gets at the same idea. So if this sketch makes sense to your brain, go ahead and use it. But it's basically saying what we just talked about from the previous picture, where you're looking at the effectors interacting with the R proteins inside the cell. Here you have the MAMPs, recognized by typically a cell membrane receptor. They're showing how the MAMPs and effectors are coming from the microbes, right? And then we have the PRR receptors, the R proteins. Okay, so different strategies, complicated immune system, which is a good thing. So these are all induced defenses. So the plant isn't like, they don't have all of this up and running all the time. It's made in defense or in response to the pathogen. And so, and then it'll kick in that signal transduction pathway. So these elicitors, the effectors, the MAMPs, those are all signals coming from the pathogen. And hopefully the plant is recognizing that. And they're doing that by those plant receptor proteins. They use those to recognize. And then they have the ability, once the elicitor gets docked into those receptor proteins, that's going to be the signal to the plant. Okay, we got to get our gene expression patterns up and running to defend ourselves. So when they looked at actually getting at the DNA level and saying what's going on at the DNA level when all of this is happening, they found that when they sequence the DNA of plants, they have found all these genes that code for these receptor proteins. So all of these PRRs and the R proteins, that, all the proteins involved in binding with something coming off the pathogen. It's huge. Those are genes. It's, when you look at all the different types of genes in the plant, it's a huge amount of genes dedicated to making these PRR proteins, the R proteins. So all of these receptor proteins, so that the plant, and that's the plant's first line of defense. It's their way of detecting who's out there, are they friend or foe? And so the plant has a huge amount of genes dedicated to making these different proteins. And again, that makes sense, especially since they're rooted in place. They cannot easily escape their predators. And so to have this level of sophistication and variety so that they can deal with whatever's coming at them, and they can have that really sophisticated, targeted response to a particular pathogen. Oops. Oh, that was weird. No, that's weird. Okay, never mind. All right, so here we're looking at another pathway here, looking at the elicitor coming off the pathogen. It looks like they drew a bacteria. So some part of the bacteria, maybe it was part of the cell wall or something like that, docked into the PRR protein. And then on this, you're seeing a whole bunch of stuff happening in response. So that's the next topic. We're going to get into some of these ideas. So here you can see reactive oxygen species start getting ramped up. And then you see in this white box, they're getting into a bunch of different responses that will kick in based on this initial interaction between the receptor protein and the elicitor. So that's the next topic I'm going to get into, but I think it's too much for just 2 minutes. So I'm going to stop here. But that's what we'll pick up on Wednesday. We'll start getting into these different strategies that the plant has. All right.